Hello everyone, I am Dr. Vineet Kumar, Assistant Professor, Department of Zoology, the Cochin College. This particular session deals with the type study in phylum Arthropoda, Pineus or Prawn. In this, we'll be dealing with the structural diversity of this particular organism, which is Pineus or Prawn. The session, as you see in this particular slide, it encompasses every system or every system which is present in this particular organism, and this will be we'll be discussing these topics in a set of 8 videos. You can see there are as per the syllabus there are 9 topics to be discussed under this type study. Of this, the last 2, reproduction and life history, will be dealt in a single video. All the others will be provided in different videos. In this particular video, we'll be talking about the external morphology, which encompasses the body features, the appendages, and also sexual dimorphism. As you can see in the slide, the particular topic which will be dealt in that particular video lecture will be highlighted. Every video will start with the same slide, and the particular topic which is being discussed in that particular video lecture will be highlighted. So in this we are going to see the external morphology. Coming to the external morphology, the shape of the body, it is more or less elongate or spindle shape, which provides low resistance to swimming, and it is bilaterally symmetrical. Coming to segmentation and body divisions, in Pineus the body is divided into 19 segments or somites bearing jointed appendages. The segments are arranged in 2 regions. As you see in this particular diagram, it is in 2 regions. The front region is called as the cephalothorax, and the posterior region is called as the abdomen. The cephalothorax encompasses the head and the thorax. It is large, unjointed, and almost cylindrical region, which has about 13 segments, but the segmentation is not visible, is not clearly visible. The 13 segments can be further divided into 5 segments of the head and 8 segments of the thoracic region. Coming to the abdomen encompasses 6 segments. As we saw in the diagram, which shows clear segmentation in each segment. The correct 6 segments can be demarcated there. There is a terminal conical piece called as the telson of the tail plate, which is not considered as a segment. The segments are dorsally round and laterally they are compressed, and each segment carries a jointed appendage, which is the swimming legs, usually called as the pleopods. Coming to the exoskeleton, which is the external covering of the body, it is made up of hard shell secreted by the underlying dermis, which is composed of a chitinous cuticle, which is being reinforced by calcium sulfate and calcium carbonate, and this strengthening or reinforcement is usually referred to as sclerotization. And the function of this exoskeleton is to provide protection, and also it acts as provisions for the attachment of muscles. And this particular the exoskeleton, as the body is segmented, there are also segmentation that can be seen in the exoskeleton, and the segments are called as scleroids. Coming into the cephalothoraxic exoskeleton, in the cephalothorax, cephalothoracic region, all the scleroids of the exoskeleton, dorsal, lateral, etc., they fuse to form a hard dorsal shield or a carapace. And the ventral scleroid also fuse to form a ventral shield. So here you can see the carapace and the structures. The carapace, also called as the dorsal shield. So this particular region, this is the entire cephalothoracic region, and this is called as the dorsal shield or the carapace. This dorsal shield or the carapace is fused with the body or body wall, the cephalothoracic body wall, and at the anterior end you can see that this particular cephalothorax is pointing, or it forms this particular structure with a serration, and that is called as the rostrum. And another thing to be noted is, as I said, the carapace is fused with the body wall, but there is an exception in the cephalothoracic region, that is towards the posterior side or towards the posterior margin of the cephalothorax. The carapace is not fused. You can see a dotted line here. From this dotted line below, the cephalothorax is not fused with the carapace is not fused with the body wall, and as a result there is a small space between the carapace and the body wall, and that space is referred to as the gill chamber. And this region of the carapace which is not fused with the body wall is referred to as a brachiostygite or the gill cover. The ventral shield at the last thoracic segment has a cup-like outgrowth, that is the same which cannot be shown diagrammatically. The ventral side of the carapace is also having fused exoskeleton, and the exoskeleton at the last thoracic segment is modified into a cup-like structure which is called as a thelicum, and it is in this cup the spermatozoa is deposited by the males. And this thelicum is seen in females, only in females, that is the ventral shield of the thoracic segment. The cup-like outgrowth can be seen only in females, thelicum is seen only in females. Males does not have that particular cup-like structure. Coming to the abdominal exoskeleton, the sclerites of each segment is separate and ring-like, and it articulates with the adjacent sclerite by a membrane, because two adjacent segments, they are fused or they are cemented by a small membrane. It is also the same chitinous cuticle, and that membranous structure is called as the arthroidal membrane. So coming to the structure of the abdominal exoskeleton. So the abdominal exoskeleton is the one that you see here. So this one is a simple diagrammatic representation that you can use for your exams. So this one is the TS of the abdomen. So here we can see that this dark or the darkly shaded region, this darkly shaded region represents the dorsal region of the exoskeleton, and that dorsal region is called as the turgum. That is this curved or arched exoskeleton which is seen in the abdominal segments is called as that is a C-shaped structure is called as it is called as the turgum, and the ventral side is called as the sternum. The ventral region is called as the sternum, and in every abdominal appendage you can see that this particular turgum, from or after reaching the body wall, it is having an overhanging. So this is the overhanging region of the turgum. This overhanging region of the turgum is referred to as pleuron, and there is a small space between the this pleuron and the turgum, and this small space, that is the black color which you see here, this is called as the epimeron. Okay, so that is the junction which is formed due to the overhanging of the turgum, which is called as the pleuron. So between the turgum and the pleuron you can see the epimeron, and the ventral exoskeleton is called as the sternum. So the region which is seen between the appendages, we can call it as this region on the ventral side, this is a sternum, and this region is called as the epimeron. So this is the TS of the abdomen. The same thing can be seen here. So this represents, so this, I have put this particular diagram to show you that this is not a single piece. As we said earlier, the exoskeleton is also segmented. You can see many segments here. So the segmented exoskeleton, to show the segmented exoskeleton, I have put this particular image because there are many pieces. Exoskeleton is also formed of many pieces, and each piece is called as the sclerite. So coming to the appendages, as we said, the every segment is having paired appendages, and a total of 19 pairs of appendages can be seen. 5 pairs of cephalic, 8 thoracic, and 6 pairs of abdominal appendages can be seen. And these appendages show a phenomenon which is called as serial homology. Homology in the sense that they are having a basic structure, but they are having different function, because the cephalic appendages are mainly concerned with feeding, the thoracic is concerned with walking, and the abdominal is concerned with swimming. So even though they are having a basic body or basic structure, they do different function, and hence they are said to be homologous organs. And because these organs are arranged in series, that is from anterior to posterior tip, they are arranged in a series, this homology is referred to as a serial homology. So the appendages are usually segmented, and most of them are biramous. Biramous means, so we'll take the basic structure of the abdomen. So we said that all the appendages are having a basic plan, and that is why it is said to be homologous. The basic plan is that every appendage will have two regions. The one region which is called as a protopodite, it is this particular region which is attached to the body wall, and a region which is which is arising from the protopodite. So every appendage will have this basic structures, and there will be modification depending upon their function. So basically, in a typical appendage, you have a protopodite with this two segmented. The segment which attaches to the body is called as coxa, and the second one where the branches arise is called as the basis. And if an appendage is having two branches, the branches are also called as rami. The two branches are an exopodite and an endopodite. If two branches are present in an appendage, the appendage is called as a biramous appendage. If at all these branches are absent or one branch is present, such a type of appendage is called as a uniramous appendage. So that's with the basic structure of this one. We talked about the branches, etc. The term endo and exo refers to the sense that the exopodite means it is the branch which is seen outside the body, and the other one which is seen towards the body. That's how the name. So this exopodite, in this case, in this particular thing, you can see that is the one which is towards the outside. This one, it faces the interior. That's why the different terminologies like exo and endo is given. Coming to the cephalic appendages, as we said, there are 5 cephalic appendages. They are the antenule, the antennae, the mandible, the first maxillae, and the second maxillae. The first maxilla and second maxillae are also called, the first maxillae are also called as maxillulae, and second maxillae are simply maxillae. So they are the other terms which are used. Coming on to the individual appendages. Of this cephalic appendages, 2 appendages are said to be uniramous. They are the mandibles and the first maxillae or the maxillulae. They are said to be uniramous appendages. The antenule is also referred by some authors to be uniramous. We'll talk about the reasons in the next session. Some authors refer the antenule as uniramous appendage. Some consider it as a biramous appendage. So we'll discuss each appendage, the structural features one by one. Coming to the antenule, it has 3 segmented protopodite. 3 segmented in the sense it is having a precoxa, a coxa, and a basis. So this is a modification from the basic plan. And 2 flagella processes arise from the basis, which are tactile in function. So here the 2 processes is not similar to the exopodite or endopodite. There are 2 processes arising from the basis, but they are not exopodite and endopodite. And because of this reason, some authors refer this as an uniramous appendage, because it has neither an endopodite or an exopodite. But some authors say that because it is having 2 branches, even though it is not exopodite and endopodite, they consider it as biramous. The precoxa has a concavity for the compound eye, and also the balancing organ called as the statocyst. Coming to the second one, the antennae. The antennae is having the normal two-segmented protopodite with the coxa and basis. As you see in this particular diagram, this is the two-segmented coxa protopodite with coxa and basis. Here the protopodite is swollen because the green glands are present in the coxa. That's why it is also called as the coxal gland. So the antenna contains the antenna mounts, or it holds the coxal gland or the green gland, and it also opens in the coxal gland through the renal aperture. The exopodite, you can see a flat fan-like structure like this. This is the exopodite, and the long one, which is segmented one, is called as the endopodite. The endopodite is having three basal segment and a long filamenta process which is heavily segmented. The endopodite is tactile in function. Coming to the third one, which is the mandibles. The mandibles, as we said, it is uniramus. Mandibles are mainly masticatory in function. It is used for cutting food. It lies on either side of the mouth. It is also having, or it has a stout calcified base and two segmented palp. It does not have a protopodite, and it does not have the rami or the branches, and hence it is considered to be uniramus. The toothed base serves as the masticatory organ. It is having the tooth, and hence it is used for cutting the food. The fourth one is the maxillulae, or the first maxillae, that is also uniramus, which is the smallest of the all appendages. It is having bilobed protopodite with coxa and basis. It is not two segmented, it is lobes. You can see here, it is not seen as segment, it is seen as lobes. That's why it is called as bilobed protopodite, and an unsegmented endopodite. Exopodite is absent, and hence it is called as an uniramus appendage. This one act as jaws, and hence they are also called as gnadobases, and they are used for pushing the food into the mouth. The fifth one is a maxillae, or the second maxillae. This is used for respiration and also for feeding. It is a last pair of cephalic appendages. It is four-lobed protopodite. It is having a bilobed coxa and a bilobed basis. Both exopodite and endopodite are flat. The endopodite is small, but you can see the exopodite forms a very broad or a flat piece-like structure, and this structure is called as the snaphognatite or the baler. And this does the function of respiration. This flat piece act as an oar, and it will be repeatedly, or it will be always, it will have a beating motion, or it always vibrates. This vibration causes the gills, or because these are present near the brachial chamber, the vibrations causes the water to flush into the gills, so that the entire brachial chamber is being supplied with a constant flow of water, and thus proper ventilation of gills occurs. That is its function in respiration. Coming to the next one, the thoracic appendages. The thoracic appendages, there are three thoracic appendages which are again used or helps in mainly it helps in feeding as well as in respiration. The other set, which is 3 plus 2, they are entirely the walking legs, and they are divided in a set of 3 and 2 because the set of 3, they are having a particular structure called as chela, or a pincer in their end, and the other one it does not have. That is why there are 3 chelate legs and there are 3 non-chelate legs. The first 3 thoracic appendages are named as the first maxilliped, the second maxilliped, and the third maxilliped. So we'll see one by one. So thoracic appendages are 8 pairs, and every thoracic appendage is biramus. They're having and every thoracic appendage is having a bilobed or a two-segmented coxa protopodite with a coxa and basis, and 5-segmented endopodite. And in every thoracic appendage you can see this 5 segments. They are the ischium, mirus, carpus, propodes, and dactylus. So we'll see that in the diagram. And in chelate appendage, as I said, the appendages, the propodes and dactylus, the last 2 segments, they form opposing, they placed opposing each other, and they form a pincer-like structure called as chela. That is why they are called as chelate legs. The exopodite is usually unsegmented. The epipodite is having the respiratory, the epipodite is the respiratory process which is connected to the coxa of maxilliped and chelate leg that is absent in the non-chelate legs. So there are 6 pairs of epipodites, 3 in the maxillipids, first, second and third maxillipids, and 3 in the 3 chelate legs. So coming to the first 3, and as I said, they are mainly involved in first 3 pairs are mainly involved in feeding, and hence they are called as food jaws or maxillipids. So we will move on to the first maxillipid. The first maxillipid is thin and flat like. And it is having 3 lobed protopodite, and hence it is gnadobasis, or it forms the jaws. It is slender and 5 segmented endopodite and an unsegmented exopodite. It is having a triangular epipodite and which is attached to the protopodite and exopodite. So this is a particular structure that can be seen here. So this one is the first maxillipid. You can see the triangular epipodite here, and you can see the protopodite, and the unsegmented exopodite is seen. The segmented endopodite can be seen here. The endopodite is normally 5 segmented. So coming to the second and third maxillae, they are having almost similar structures. There are 2 segmented protopodite, 5 segmented endopodite, and an unsegmented exopodite is seen. They are having a special, from here the structure of the epipodite is a biforked, or in the form of a fork, and that is attached to the coxa. Only in the first maxillipid you are having a triangular epipodite. In the rest, the epipodite is in the form of a fork, which is bifid. Coming to the walking legs of periopods, there are 3 pairs of chelate legs and 2 pairs of non-chelate legs. Chelate legs are having 2 segmented, as you see here. As you see in the diagram, they are having 2 segmented. They have a 2 segmented protopodite here. In the coxa, you can see here the epipodite. A small unsegmented exopodite can be seen. The endopodite, you can see it is 5 segmented with ischium, merus, carpus, propodus and dactylus. You can see here propodus and dactylus, they form a pincer-like structure. That is why they are called as chelate legs. And mainly the chelate legs helps in taking the grasping the food and pushing it into the mouth, and also it is used for attack. During attack, it is also used as a weapon. The non-chelate legs or the non-chelate periopods. Periopods means walking legs. There are 2 pairs. They are also having 2 segmented protopodite, 5 segmented endopodite, and an unsegmented exopodite. Epipodite is absent here. Here you can see the dactylus and propodus, they do not form the chela here. They are seen one behind the other, propodus and the last segment, which is called as the dactylus. That is why they are called as non-chelate legs. So here the dactylus is immovably articulated to the propodus in a straight line. The other one is movable. It is a pincer because that is used to grasp the food particles, attack the enemies, etc. So that's with the thoracic appendages. We have discussed the 8 thoracic appendages, 3 maxillipids, 3 chelate legs, and 2 pairs of non-chelate legs, which makes the 8 pairs of thoracic appendages. Coming to the abdominal appendages, the abdominal appendages, there are some specialities with the abdominal appendages. The first abdominal appendages of male and female is different. Second to fifth is the typical appendage, which we discussed earlier, but with a basic plan, which we see from 2 to 5. The second to fifth will have the basic structure with 2 segmented protopodite, an exopodite, and an endopodite, which are unsegmented exopodite and an unsegmented endopodite. And in male and female, the first one is different, and the last one, the sixth one, is also different. That is also called as the appendage is called as a uropod. So 2 to 5, it is the typical appendage, and these appendages of the abdomen, or every appendage, it is used for swimming, and they are also called as pleopods. They are swimming appendages. So abdominal appendages, there are 6 pairs, and all the appendages are biramous, used for swimming. 2 segmented protopodite, unsegmented endopodite, and exopodite. So the first appendage in female is more or less like the typical appendage. The difference is that, as we see here, the only difference in females first appendage is that you can see here the endopodite is shorter, the exopodite is long. But in a typical one, both the endopodite and exopodite are having somewhat the same size. But in females, you can see here the endopodite is smaller than that of the exopodite. And coming to the case with males, as we see here, in the case of males, the first appendage is special in the sense, in males, the endopodite of the first abdominal appendage is having many setae. In both sides, they are having setae, and these the setae, they interlock and they form a tube-like structure. So here you can see this is this tube-like structure. You can see the endopodite of two sides. They form an interlock-like structure which forms a tube-like structure, and this tube-like structure is called as the patasma, and this tube is used to transfer the spermatozoa from the males to the female thelecum. So this is a special structure that is seen in the abdominal appendage, first abdominal appendage of males. As we said, female does not have any other specialities other than the endopodite is small. Here the endopodite is having many serrations or setae. With that, the endopodite of both sides, they form an interlocking structure, a tubular structure called as a patasma, which is used to transfer the sperms. And coming to the, so that was the first abdominal appendage. Then coming to the another special one, which is called as a uropode, which is the last abdominal appendage. Here the coxa and bases fuse to form a structure which is called as a sympode, so here you can see. So the coxa and bases, they are here the coxa and bases. They fuse to form a structure which is called as sympode, and the exopodite and endopodite is flattened, and it forms, or it has a shape of an oar. And this particular structure called as a uropode, that is used for backward movement or backward swimming. So these appendages are used for mainly used for swimming. They are swimming appendages or pleopods. So that's with the abdominal appendages. So we have completed the appendages. We saw the five cephalic appendages, 8 pairs of thoracic appendages, and the 6 pairs of abdominal appendages. So next is, you can see these videos. So here you can see for walking, these organisms, they are using their thoracic legs for walking. And in this particular video, so here in this video, you can see for walking, they are using their thoracic legs. And in this one, you can see when they swim, when one moves here, you can see for swimming, they are using the abdominal, you can see here, they are using the abdominal appendages for swimming. In that time, the thoracic appendages, they do not work. So this is regarding their appendages. And coming to the sexual dimorphism, which we have discussed here, males are smaller, females are larger. The patasma is a structure present in males used for transferring the sperms. Patasma absent in females. Thelecum is absent in males, but thelecum is present in females, which is a cup-like structure of the formed in the last ventral side of the last thoracic segment, which is used for receiving the sperms. The genital opening is seen on the coxa of the last pair of walking legs. In the females, the genital opening is seen on the coxa of the third pair of walking legs. So this much about this particular session. So we'll come up with the next session, which will be the digestive system. So hope this is clear, the appendages as well as the sexual dimorphism. Thank you.